Thank you for standing by and welcome to the Imugene Investor Webinar, which forms part of the company's non-deal roadshow this week. Joining us from Imugene today, we have the CEO and Managing Director, Leslie Chong, COO, Bradley Glover, and Chief Medical Officer, Dr. Paul Woodard. All participants are in a listen-only mode. There'll be an initial slide presentation lasting for around 20 minutes, and we'll take questions thereafter. Some questions have been submitted via email, and if you'd like to submit a question via Zoom, please do so using the Q&A panel within the Zoom app. We've already received quite a few on email, but we'll get through as many as we can in the time permitted. To kick it off, I'll hand it over to Leslie. Please go ahead.
Thank you, Matt. It's a pleasure and honor to talk about Imugene, and I have my dear colleagues here with me in Australia to help me go over these slides. So first of all, Imugene is listed on the ASX and the ticker is IMU. We are developing transformative cancer medicine for patients. My disclaimer. So investment highlights as a snapshot. Cash on hand as of September 30th, we have 163 million. On this presentation, we will focus on our three prioritized platform technologies, and that's allogeneic CAR T cell therapy, our onCARlytics, our CF33 oncolytic virus, especially the MASS study. We have focused on 5 clinical studies that we'll be very focused on and moving forward with. And I'm proud to say we have an in-house GMP cellular therapy manufacturing facility in North Carolina. We are addressing many cancer types and solid tumors as well as blood cancers, and we have a long life patent to get us there. So our clinical executive team has over 150 years of combined experience, and not only in clinical development, but also in drug approval. So all of us have roughly around 13 FDA approved drugs that we've taken to market. And this is not, is specifically focused on cancer medication. We have key catalysts coming up in the next 12 months with Azer-cel already meeting the key milestones of first patient in on our phase 1b confirmatory study. Then we'll have lots of enrollment updates thereof, and by the end of the 10 patient enrollment, we'll start our conversation with the regulatory authorities to start our registrational phase 2 market study. Our onCARlytics, we already have a patient in, and we're also looking to enroll a few more patients just in the next quarter. That onCARlytics will have first patient in the combination and IT and IV and lots of news throughout. Vaccinia will continue to have readouts. We just noted a recent status update on data that we're very pleased to talk about here, and we'll have more news throughout, and also onCARlytics and Azer-cel combination to address a great need in solid tumors. Our commercialization strategy is quite simple. Our clinical readouts and success really drives that value realization opportunities, and that is company acquisition. And this is not an acquisition of another company, but this is Imugene's acquisition or future acquisition. Partnering with big pharma. We want to license out our technologies separately, and also with the Azer-cel, it expedites the potential of developing and commercializing independently. We have 4 unique platforms, but today we're really going to focus on our 3 prioritized platforms. And I am going to switch it over to Dr. Bradley Glover to talk about our exciting cellular therapy program, Azer-cel.
Thanks so much, Leslie, and thank you, everyone. So let's go ahead and get started here. So I'd like to start first by talking a little bit about autologous CAR T therapy. So CAR T is an acronym for chimeric antigen receptor T cell. Autologous CAR T therapy is a cancer treatment which involves taking a cancer patient's own blood and sending that to a manufacturing site or a processing lab. These patient T cells are then re-engineered to recognize cancer cells, and then they're infused back into the cancer patient after that. There are currently about 6 approved autologous CAR T drugs for blood cancers at this current time. So what is allogeneic CAR T therapy? You may have heard it called auto or allo. This is the allo version. It's actually a living drug, and it's off the shelf. Allo CAR T is a type of immunotherapy that uses healthy donor T cells that are modified and engineered to be used off the shelf. What that means is that the healthy donors are the ones providing the initial starting material, which has a lot of benefits we'll talk about on the next slide. These are processed, and each batch can make multiple product doses for patients. And these are extremely effective in killing cells, in this case, expressing the CD19. Next slide. So a few key differences between these two types of therapies, between auto, as we talked about, coming from the cancer patient's own cells, and allogeneic coming from a healthy donor. So auto CAR Ts are made from those persons, the cancer patient cells, whereas the allo CAR T come from a universal donor, and that means broad patient access. The manufacturing times for autologous CAR T manufacturing can be weeks to over a month. That means these cancer patients are waiting to get their therapy and sometimes even have to undergo extra chemotherapy while they're waiting for their treatment. On the other hand, allogeneic cell therapy, these cells are pre-produced from healthy donors. They're engineered, they're ready when the patients need them. They're faster, they're cheaper, they're frozen, and we can distribute those immediately when a patient needs them. And that there is also an additional benefit. Because you're using these healthy donor cells, these are extremely potent, and the variability, a lot of it has been driven out of the process. As we mentioned a second ago, there are currently 6 FDA-approved auto CAR T drugs in blood cancers, but at this time, there are no allo CAR Ts yet available approved by the FDA. Next slide. So we really believe that Imugene is an industry leader in allogeneic cell therapy. We acquired azercaptidine zapralucel, what we call Azer-cel, in August of 2023. Phase 1 trial was completed with 84 blood cancer patients with promising safety and efficacy data. The patients were in DLBCL who relapsed after autologous CAR T therapy. They demonstrated an 83% overall response rate, a 61% complete response rate, and duration of 55% duration at greater than 6 months. We've received positive feedback from the FDA on our results already, and we're undergoing a phase 1B confirmatory study to enroll an additional set of patients that have relapsed after their initial auto CAR T therapy. And we're excited, and we've released this. Our first patient has been dosed, first patient in on November 10th of 2023, not too long ago. And our strategy is to commence our registrational study, our phase 2 study, in the next 12 to 18 months. You may ask, what is the size of the market? The market is significant. It's $2.5 billion per annum for DLBCL CAR T relapse patients. It's important to understand that 60 to 65% of patients currently treated with autologous CAR T therapy are going to relapse, unfortunately. And by 2025, the relapse market is going to continue to grow. So once the patients have that relapse, Azer-cel could be potentially available to address that market need. So I just mentioned our phase 2 potential registrational trial. We expect it to begin after our phase 1B study in the second half of 2024. The population is for DLBCL patients who have relapsed after auto CAR T therapy. We've received FDA guidance on the potential registrational study. We did that. We had that conversation in July of 2023. We have over 35 sites in the U.S. for our phase 1B trial, including Moffitt, City of Hope, and the others listed here. The drug product for our phase 1B has already been manufactured and is frozen and ready to be distributed to patients. Our drug is manufactured at our manufacturing site in North Carolina. I'll talk a little bit about our manufacturing process. As we mentioned, we receive these blood leukopaks or leukapheresis from healthy donors. These T cells within there from the healthy donor are engineered. We knock out the TCR, we knock in the CAR T. Basically, it's a surface receptor that helps the T cell find and locate cancer cells and destroy them. These cells are then expanded and frozen down in these little vials. And this is possible because we have the infrastructure, we have the manufacturing and the gene editing technology and the process understanding, and all of it is done with a significant amount of quality systems. And here's a little snapshot of our manufacturing capabilities. So we have laboratory space that's dedicated to research, process development in North Carolina. This really enables us to have a continuum of going from research to process development and then to our manufacturing site that's shown here on the right. We call it MCAT, which is Manufacturing Center for Advanced Therapeutics. This is a fully operational GMP manufacturing facility, and it has already manufactured product that is in patients currently. We have a significant amount of capacity and to meet patient demand. We have a highly skilled workforce, and it really is a competitive advantage we feel here at Imugene because it's de-risked the CMC process all the way through that a lot of companies are facing challenges with. And at this time, I'd like to hand it over to Dr. Paul Woodard to discuss the rest of our programs.
Thank you, Brad. I'm going to talk about our two oncolytic virus programs. First is CF33, our vaccinia program. CF33 can infect and selectively kill cancer cells. If we look at the cartoon shown here, when the virus is either directly injected into tumors or infused intravenously, the CF33 virus enters the cancer cells, duplicates itself, and then causes the cell to rupture or explode, which releases new virus particles capable of infecting other tumor cells. It's an engineered virus closely related to smallpox that doesn't exist in nature. It was invented by Professor Yuman Fong at the City of Hope and can be further modified to carry payloads to increase killing. And there are multiple ways that it's capable of killing cancer cells. It can directly kill cancer cells through infecting them and lysing them, can activate the patient's own immune cells to kill cancer cells. And when the tumor cells rupture, the particles from the tumor can prime the immune system to further enhance the immune response. There are several precedents for oncolytic viruses that have already been approved. Next slide. We have a phase 1 study that's been making a meaningful impact for patients. This study is treating advanced cancer patients either intravenously or intratumorally. There are two parallel cohorts with the CF33 virus alone, or in separate cohorts in combination with pembrolizumab, an anti-PD-1 antibody in multiple different solid cancers. 34 patients have been heavily pretreated patients have been dosed to date. The doses have been determined to be safe and tolerable. There have been no dose-limiting toxicities. And 25 patients are currently evaluable for response. That means they've received treatment and they've had their first scan at 6 weeks. And we have one patient with a complete response in bile duct cancer, one partial response in melanoma, and both of these are at a middle dose level, and 16 patients with stable disease. I'd like to expand a little bit on the patient with a complete response. This is a patient with bile duct cancer who had 3 prior different types of chemotherapies and had no response to any of them. This patient came on study, received CF33 intratumoral injection, and achieved a complete response and has been on our study for more than a year and has been off treatment for more than 200 days. In 7 patients with some of the hardest tumors to treat, gastrointestinal cancers, who received CF33 alone, we've seen a stable disease, partial response, or complete response rate that's called disease control of 86%, which we think is very encouraging. The study expansion is planned for 10 additional patients with bile duct cancer. This phase 1 trial is being conducted at 12 centers in the US and in Australia. Next slide. I'd next like to talk about our next oncolytic virus program, which is CF33 CD19. We call it oncolytics for solid tumors. So what is this and what is different than CF33 by itself, and why CD19? So the backbone of oncolytics is the same CF33 virus, but it's been engineered with CD19, which is a protein it causes expression of a protein called CD19 on the surface of solid tumor cells. Now, many blood cancers like leukemias or lymphomas have this protein CD19 on the surface of their cells. And when you modify a patient's T cells to see the CD19 signal, the T cell really targets those tumors that express CD19. So if we use this CF33 CD19 virus, we can cause solid tumor cells to show the CD19 protein on the surface, and then we can treat patients with CD19 therapies. And we're targeting tumors like breast cancer, lung cancer, gastric cancer, colon cancer, and none of these normally express CD19 on the surface. Really, the holy grail in CAR T therapy is to find a way to make it work in solid tumors. This technology, Imugene's oncolytics technology, seeks to overcome the challenge in solid cancers and make solid cancers visible and vulnerable to CD19 targeted therapies. Next slide. So how does this work? The CD19 or CF33 CD19 oncolytic virus makes solid tumors seen by CD19 targeting therapies. Oncolytics infects cancer cells. It then reproduces inside the cells and causes the cell in panel 3 to express CD19 on the surface, and then you can target it with CD19 therapies. Those cells then die and release viral particles that can infect additional tumor cells. Next slide. So we've treated the first patient on our phase 1 OASIS study. These are patients with advanced metastatic solid tumors. And this study is designed to look at oncolytics by itself or in combination with a drug called Blincyto, which is a bispecific antibody that targets CD19. Patients are treated either intravenously or through direct intratumoral injections into their tumors. We enrolled the first patient, which is a patient with ovarian cancer, at City of Hope in October. We have a phase 1 study, or this phase 1 study is planned for around 10 sites in the US. And it's worth saying that there are many different CD19 approved drugs, which could become the preferred partners to combine with oncolytics. Next slide. What we'd ultimately like to do is combine oncolytics with our own internal azer-cel program, which is a CD19 targeted allogeneic CAR T program, in multiple tumor types in patients with solid cancers. So we do have some early preclinical studies ongoing where we combine azer-cel with oncolytics in different solid tumor types. And what's very impressive about this, so you combine the cells with these two drugs, and we saw 100% killing of triple negative breast cancer and gastric cancer cell lines. And I've done a lot of these sorts of studies and seen a lot of these sorts of studies. It's very rare to see 100% killing of any tumor type in vitro studies. So we're very excited about these results and intend to put this combination in the clinic later on. Next slide. So I will hand it back over to Leslie. Thank you.
Thank you, Dr. Woodard, and thank you, Dr. Glover. So, in the last few months, we have seen a high volume turnover. The 6 months average there was around 800 shares, and it's gone up just in October to 854 million. It will be interesting to see how we do for November. But our top 10 shareholders consist of Paul Hopper and myself, in addition to the Vanguard Group, BlackRock, UBS, and others. And our directors and management group own over 6.5%. of the company, which is nice to see. We've included an acronym, a glossary, because those of you who've asked us to include this, and we've done so you can follow along. And that is the end of the presentation. So we'd like to open the floor up for some questions, please.
Thanks, Leslie, and here to Paul and Bradley. As you mentioned, we'll now move on to the Q&A session. Once again, if you have a live question, please feel free to type it in using the Q&A panel within Zoom. But we'll start with some that came through via email. The first question I have is, could you please elaborate on the synergies between azer-cel and the oncolytic CF33 program?
Sure, I can
That's
take
an
that.
easy one, I would say. Go ahead, Paul.
So the oncolytic virus kills tumor cells by itself. If you then combine expression, cause the tumor cells to express CD19, you can use a CD19 targeting agent like azer-cel to kill tumor cells expressing CD19. So you have sort of multiple opportunities to kill solid tumor cells through the virus and through the CD19 molecule.
Thank you, Paul. The next question is, will the azer-cel phase 2 registrational study be completed first before combining it with oncolytics?
We're going to do that as fast as we can simultaneously. So we do not have to wait until azer-cel, our blood cancer program, to be completed before we start the combination.
Thank you. Moving on to the vaccinia, has the process started of applying for breakthrough therapy?
So that is material information. So I won't comment on that. However, the process is quite lengthy. You have to get a lot of preclinical as well as clinical data, put that together to the FDA, and once you submit, it's a 60-day process in order for them to come back. That's just the process. I'm not stating anything but the process at the moment.
Thanks, Leslie. The next question is, the recent vaccinia data was cut in time. Does that mean the results can still further develop and potentially turn some stable disease into partial responses or complete responses? And following on from that, what is the timeline on hearing about the other 7 patients awaiting further scans?
So the data cuts are, it's a lengthy process where the information is entered into what's called a case report form. So the case report form is what inevitably gets to the FDA. So that document has to be the truth. It has to be evaluated against a medical chart. There is a professional that has to actually go out to the hospitals and clinic and check and verify. And then in addition to that, that gets entered into a database. Then it's statistically analyzed. Then Dr. Woodard has to go through it with a fine-tooth comb along with his clinical scientist before it's ever published. And so it is a lengthy process, and we only want to do that when it's necessary for a submission into a major cancer conferences and things like that. So we will provide updates. However, I just want everyone to understand that that process can be quite interruptive to the program, but also it's done when we want to submit a statistical, analyzed, validated information to a major conference. And ergo, that's why we were able to publish on our recent data, because we were putting that together for publications and abstracts and of such.
Thank you. The next question is, Imugene announced last week the expansion into bile duct cancer. Is this expansion actually a phase 1b trial, and does this extension impact the previously reported estimated completion of May 2024 for the existing phase 1 trial? And following on from that, could management please provide an estimate of the additional time required for the trial?
So the reason why we're dose escalating in the MASS study is because we are seeing no safety or toxicity issues, and we're seeing responses, which is, beautiful, and we want to get up to as high as we can. So that takes time. You have to enroll 3 patients and then have to wait a month to evaluate whether this is safe, etc. The the expansion of the bile duct cancer will be done within the phase 1. It will not be a separate study unless we see something profound within those 10 patients that we think that we could expedite the process in another, perhaps even a phase 2 study. So it is a pretty exciting expansion for us. However, we're going to be very careful and make the right selection of the dose and the timing. So I can't say for sure if the May 2024 timeline will move, but I think as we explore more doses and more response rates, then we want to be able to enroll those patients in a very steady but yet careful manner. So that time may move, but at this time, we don't know. But it's a good thing.
Leslie, the next question is, can you please advise what the outcome of featuring CF33 at CHEAC was in early November? It appears that this was not the conference where the biopsies mentioned by Professor Yuman Fong a few months ago were accepted for analysis at a major conference. Can you confirm these biopsies remain scheduled for an upcoming conference?
I'm not sure what the biopsies were relating to. I don't really recall. Biopsies are sample analysis of certain protein types on the cancer cells. So I'm not sure, but the CHEAC was a trial in progress submission. We actually didn't have this data until after the submission deadline. So you generally have to submit an abstract months before the event and get accepted. So the fact that we had the data after the submission deadline, yeah, I'm not sure what the biopsies. Someone can write to me and clarify what they mean by that question.
Problem. The next question is, are patients who were dosed at low to medium levels with vaccinia, are they continuing to be treated at higher levels as the trial progresses?
No, so a dose escalation and dose finding studies, you don't want to convolute various different doses by dose escalating. Sometimes in a phase 1 study, you do allow intra-dose escalation, but for this, we want to be very clear that a certain dose is working. And so to move a low dose person into a high dose, you then alter that data. So we do not allow this at this time for this study.
There's been several questions around the status of CHEAC. Can you provide an update on what the latest is there?
So CHEAC study is currently in cohort 4. We think that CHEAC will start competing with the MASS study. And in addition to that, Dr. Yuman Fong, along with City of Hope, will fund that study by themselves. So we don't have to, Imugene as it were, we still own CHEAC. However, we don't have to utilize the resources nor the funding to increase the dose at that study. So they will provide alternative financial resources to continue on with that indication or other indication.
Thank you. A question more on the corporate side. Would a potential NASDAQ listing be on the cards, especially if accompanied by big news, to access more sophisticated investors overseas and to further bring attention to Imugene's technologies?
The simple answer is yes.
Easy. Excuse me, the next question I have is there any chance Azer-cel could get FDA accelerated approval allowing Imugene to market the drug subject to a parallel confirmatory trial?
So yes. So this is one of the reasons why we're doing the confirmatory study in auto CAR T failures in the 10 patients with DLBCL, because that group of data will provide evidence of the registrational study that we'll go into, and it depends on the design of that study before we can ask for or look at accelerated approval, but I think that's actually a possibility for us.
Thank you. The next question I have is, for the one complete response in bile duct cancer, how many times was the patient injected with CF33, or is the study a one-time injection and then monitor the patient?
Paul?
It's a one-time injection. So this patient had a single injection of CF33 and achieved a CR.
I mean, that really speaks to the power of our oncolytic virus, that the patient received not multiple doses, but a couple of doses, I believe, and then went on to, for the last 200 days or more, having a complete response. So the immune system is turned on and it's doing what we have promoted the virus to do.
Thank you. And along the same lines, the next question is, would the goal be to move to a registrational phase 2 trial if the bile duct expansion trial is deemed a success?
Anything can happen. I mean, with response rates, you can have a meaningful conversation with the FDA right away on various different things. So yes, we hope for, that's one of the reasons why we're expanding into the 10 patients in bile duct cancers.
Thank you. I'm going to assume this is with regards to one of our recent azer-cel announcements, but the question is, what does positive feedback from the FDA actually mean?
That you've had a dialogue with them, there's formal and informal written documentation to go forward with certain kinds of studies, and then you have you want an open dialogue with the FDA so that when you apply for that phase 2 registrational study, they're already consenting to it. And so you can run your study with the confidence of knowing that the FDA will approve either an accelerated approval or an approval down the line. So that's what we have.
Thank you. Coming back to the comment around the potential acquisition of the company, can you shed any light on who would be a natural acquirer in terms of hypothetically company names?
That would be a little bit material for us, so I think I'm not going to comment here. Yeah.
Problem. The next question is, what clinical trials will you do in Australia and where? I know we've already got a couple of sites for Vaccinia, but maybe you can expand on that a bit.
So the MASS study is already here, and Dr. Woodard and our head of ClinOps went to go and visit some Australian hospitals and clinics, and they're quite keen to join our study, so we may have more. We also want to bring azer-cel because there's auto CAR-Ts approved here, and when those patients fall off of those therapies, they don't really have anything, so we'd love to offer that to Australian patients. So azer-cel will likely come here for the registration part of the study, as well as the expanding into the MASS. We're also looking at onCARlytics to come to Australia as well.
The next question is, if we are limited to 1,500 doses per annum with the current manufacturing facility, how fast can we scale this up, assuming approval?
Brad?
You bet. So the way in which you address the capacity, I mean, there are several types of capacity, operational, infrastructure related, employed related. When we talk about the 1,500 doses per year that are available, that can easily be flexed depending on the number of shifts that you run, the number of suites, and we have a significant amount of opportunity to be able to expand and almost double our size into a shell space that is part of the facility. So if new technology comes along, if production starts, or if demand starts to ramp up, we have ample lead time to expand our capability or run the facility at a higher level of capacity. It's not a problem.
And another question for you, Brad, while you're at it, is for patient-acquired blood, is the process similar to plasma extraction regarding the time taken for the patient?
Well, so the leukapheresis process, we have vendors that we work with and identify donors. They go to the facility, they participate in the leukapheresis process, and then that is shipped to us fresh, and then we engage in the processing and isolation of the T cells.
Thank you. This is a follow-up question from earlier, but is it possible that some of the stable disease patients on MAST will improve to partial response or even complete response?
I'm going to let Paul talk about pseudo progression. I think that will help answer that question.
Yes, that's possible. So one thing that happens with immunotherapy sometimes is a phenomenon called pseudo progression. It actually looks like on a CT scan that the tumor has grown or progressed. And what that sometimes represents is immune cells infiltrating the tumor, which is what you want to happen. And then the tumor subsequently shrinks. And that's what happened with our complete response patient. It looked like their tumor had progressed. And then 2 scans later, I think they were in a complete response. So this does happen with immunotherapies at times. But absolutely, there's a chance that other stable disease patients could convert to responders. And I should also say that we're continuing to enroll patients. So it's a moving target. You know, we had 34 patients when we did this data cut. We have more now.
Leslie, there's been a couple of questions with regards to HER-Vaxx. So just in general, can you give an update on the status of HER-Vaxx?
Yes, so HER-Vaxx has moved right along, and we will be focused on the next Horizon study. This is the third, fourth line in the gastric setting, because we think that if we have success there, then we could even put it towards a registrational study. So all our programs has been reprioritized for the next data readout or earlier data readouts, as well as a registrational path. So we want to focus on things that's going to give us data, be a benefit for patients, and hopefully get a registrational path early on.
Another question that's come through is the bile duct cancer complete response patient off limit to interviews or testimonials with regards to their experience?
Patients have to consent. Normally, a lot of patients with cancer, they don't necessarily want to broadcast that they have cancer. They want to live their lives. So we generally don't. And then there's a confidential nature of patients participating in clinical trials. So we generally don't approach them. We'd like for them to live a healthy, normal life without cancer. So we generally don't bother them.
Thank you. We've just got time for 1 or 2 more. So is there any relationship between Amgen and Imugene with regards to Blincyto? Is it being purchased and trialed entirely independent of them for oncolytics?
We have purchased the Blincyto. We continue to have conversations with various different pharmaceutical companies, but if you purchase the items yourself, you have a lot more control. And I'll just say that.
And the final question, Leslie, is just what's the latest with regards to PD-1 Vax?
So PD-1 Vax is currently in the Imprinter study, which is a lung study, non-small cell lung study. We will be coming back with news very shortly on that. So we'll be coming back with some exciting news on that.
Thanks, Leslie, and to you too, Brad and Paul. Just to wrap up, there was also a question around whether these slides today were available. They are on the Imugene website. If anyone has any further questions that they'd like answered, please feel free to reach out via the shareholder inquiries email address, which is available at the bottom of all the announcements. The webinar's had a huge response. There's been about 400 people on live and a lot of questions come in, so we can't cover absolutely everything, but we really appreciate everyone's interest, and thanks for your time today, and thanks again to Leslie, Paul and Brad for your efforts.
Thank you. Bye all.
Thank you. Bye-bye.
Too.